All right, yeah, it's my pleasure to introduce Saroj Poudel. Saroj is a NASA astrobiology postdoc. He's been working with the ENIGMA project since 2018, and he's got his PhD from Montana State, where he worked with Eric Boyd on the phylogenetic analysis of microbial evolution. He's, as everybody in the team knows, he's very collaborative and sought after by not just people within the team, but also by outside collaborators. Because of his impressive aptitude in informatics and phylogenetics, and up to now, he's author on over 30 publications. So Saroj is going to discuss his recent work on deconstructing the molecular evolution of Rubisco. All right, Saroj, thank you.
Thank you, Vic. Okay, first of all, thank you to all the speakers for a wonderful talk, and thank you all colleagues for doing the meeting and for all the wonderful questions. Okay, so I'm gonna talk about Rubisco that I did past year. And I'm going to keep it short because this work has already been published. So, right. So a refresher on Rubisco. So the main function of Rubisco is to fix carbon dioxide, right? And you might have often heard Rubisco being the most abundant enzyme, right? It's because most of the photosynthetic organisms, they rely on this Kelvin cycle that where Rubisco is the key enzyme to fix the carbon dioxide, right? And Rubisco being one of the most inefficient enzymes, they have to really produce a lot of it, which is why it's the most abundant enzyme on Earth. And the reason that it's really inefficient is because it cannot discriminate between two of its substrate, carbon dioxide and oxygen. So when it accepts carbon dioxide, it's really happy, it makes sugar, right? And when it accepts oxygen, it eventually releases carbon dioxide. So thereby decreasing its efficiency. And in fact, more than 30% of the time in higher plants, it accepts oxygen instead of carbon dioxide, which is why it really decreases its efficiency to be able to one of the enzymes fixing carbon dioxide. So the main question of this project was, can we look at a sequence and structural features and identify the key distinct features in these enzymes that can allow them to bind CO2 over oxygen. And we're gonna investigate using specificity ratio. So using the equation as shown here. So broadly speaking, specificity allows us to basically identify enzymes that can select carbon dioxide over oxygen, right? So higher the specificity score, it basically means that enzyme can bias CO2 over O2, and lower the specificity ratio, that means that you can't discriminate carbon dioxide to oxygen. So to date, there are 4 types of Rubisco, form 1, 2, 3, and 4. So form 1, it has two subunits, large and small subunits, and they become an octamer, meaning 8 subunits. So 8 large subunit and 8 small subunits. So they're found in all plants, cyanobacteria, and so on. And form 2, which is the simplest form of Rubisco, so it's dimeric, and it only contains a large subunit, so it does not have a small subunit. It's found in protobacteria and methanogens. And form 3, it's found in methanogens and Acrinearchaeota, which is a phylum in Archaea. And the structure varies. It comes in dimeric or pentameric, so 5 subunits, right? And lastly, form 4. So structurally, it's similar to the Rubisco, but we don't really know the function yet. So the first thing I did was to reconstruct the phylogenetic tree of Rubisco, right? So for those of you who are not aware of phylogenetic tree, so basically there are 2 types of trees that we can recreate, rooted and unrooted. So in this case, I've rooted this tree to form 4. So remember, form 4, structurally it's similar, but functionally it's not different. It's different, right? We don't know the function yet. So by rooting this, what that means is we know the starting point, right? So the first divergence is where we can say that this is the ancestor of Rubisco and Rubisco-like proteins. So the next divergence point is where the ancestor of this modern Rubisco is. So as you move from the root to the tip, you're basically moving along the time, right? So all these organisms that I have is homologs of Rubisco. meaning the different Rubisco found in these organisms. So we have a total of 54 homologs in here. So the red star basically means that we have a solved crystal structure. And so there are two columns that I've overlaid the information here. So the first column is the specificity. So the dark blue are the Rubisco that has a low specificity, so basically less than 50. And then if you go all the way to 1D, it's higher specificity, right? So again, remember, as I was saying, you go from the root to the tip, you're moving along of the time, right? So the first divergence from the ancestor is group 2. So what that means is group 2 is much closer to the ancestral Rubisco, right? And group 3 emerges, and then group 1. So basically 2 being the most ancestral and 1D being the most modern, right? So the take-home message in here is if you compare group 2 and group 1D, you can identify or detect some of the features that has allowed 1D to select CO2 over group 2 Rubiscos, right? Because remember, and looking at this data, all of the group 2, the specificity is very low, meaning they can't discriminate between CO2 and oxygen, as opposed to group 1D, where specificity is really high, somewhere from 70 and above 90. So looking at the distribution again, showing the same result as in the 2003, this group 3 only had 1 Rubisco, so I excluded from the data in here. So the x-axis is the different groups, y-axis is the specificity. Again, 1A, looking at all the subspecies of group 1, they have really high specificity compared to group 2, and they are significant as well, right? So again, if we can compare group 1D and then 2, we can identify the features that has allowed 1D to select CO2 over oxygen. So the first thing we did was looked at the charge distribution on the surface of the Rubisco. So I took 3 proteins, 3 Rubisco homologs, 5-MAC, which has the lowest specificity on the left, and then 1BXN, which has the mid-range specificity, and 4F0K, which has the highest specificity, right? So the red color here means negatively charged residues, white, the neutral, and then the blue, the positively charged residues. So if you look at 5-MAC, the distribution is almost negative to neutral, right? And if you move to the mid-range Rubisco, 1BXN, so then you start seeing this positive tunnel, right, in the middle. So at the bottom is from the side view, and the top is from the top view. So you start seeing this positive channel that's connecting to the active site. Well, I wish I had shown the active site in here. But and if you go to 4F0K, that tunnel is more developed, right? So meaning that it looks that these tunnels, as they're going from lowest specificity Rubisco to highest specificity Rubisco, they are selecting for these positive residues to make this tunnel that can connect the active site. And thinking about it, or taking a closer look at this enzyme and then removing all the neutral and negatively charged residues and only focusing on this positive tunnel again. So we start looking from the active site. So the red circle here is highlighting the active site. So looking at 5-MAC, the positive tunnel is really small, right? And then if you go to 1BXN, it increases. And then as you move to 4F0K, it's really developed, right? So it almost connects to the surface, and it's connecting the 2 active sites. So now the question is, how are the Rubisco really benefiting from this positive tunnel, right? So if you think about the 2 substrates that it accepts, oxygen and carbon dioxide, both don't have any net charge. But what carbon dioxide has is this quadruple moment. So that charge distribution, it's attracting this positive residues, right? So it's not that oxygen can go through this channel, but every time it sees carbon dioxide, it's pulling towards the tunnel, right? So they can guide to the active site. So that allows this Rubisco to select CO2 over O2. So we see this from only these 3 structures, right? But looking at the 2003, we had roughly 10 to 11 structures, right? So are these really correlated? So shown here is a correlation plot. Again, x-axis is specificity, and y-axis, I'm looking at the surface area of the cavity, right? So if you move, and then what we see is a really strong correlation of 0.6, meaning it's explaining the 60% of the variance, right? So basically, if you move from the lowest specificity to the highest specificity, the volume increases. So it's a really strong indication that one of the features these modern Rubisco is using is this cavity to select CO2 over O2, right? But again, what this is we're just looking at 10 structures. But remember, I have 54 homologs, right? So are these rubisco that we find in group 1D, are they selecting or evolutionarily selecting these residues? So again, I'm looking at, again, comparing group 2 and then 1D. So group 2, 5-MAC, remember it has the lowest specificity. So what I did was I looked at the residues in 5-MAC that form this active positive tunnel, and then looked at sequence conservation. So red here is greater than 90% conserved, and cyan here is less than 30% conserved. So at 5-MAC, it's really conserved only around the active site, right? And then as you move or radiate out from the active site, you start seeing these residues or these positions where these amino acids are not really conserved. You go to 1D, which has the highest specificity, the active site, and even when you radiate out from the active site, all these positions are really conserved, right? Now you go to 1D, remember 1D had really developed tunnel, right? So if you take all these positions, residues and all these positions, and go into 1D homologs and then see for sequence conservation, you see that these residues are really conserved. What that means is you can pick any homologs in group 1D and you would see that positive residue in that position. But if you go to group 2 homologs where they have the lowest specificity and you look for sequence conservation, beside the active site, these residues are not really conserved, right? So what this says is basically as the rubisco are evolving, they're selecting for these positive residue that can make this positive tunnel. So with that, thank you to Paul and Vic for guiding me through this project. And special thanks to Ross for bringing this project to us. And thank you everyone involved in this great project. And with that, I'm happy to take any questions.